So today we're going to talk about SiRNA gene silencing mechanism. Firstly, suppose that there's a cell. This is a cell, and sorry, just ignore my drawing. I'm not that an expert in drawing. So now you have a double-stranded. This is a double-stranded RNA. Suppose, all right. This is a double-stranded RNA which enters the cell. So cell. Okay, so double-stranded RNA, which after entering the cell, it gets chopped into smaller pieces. Suppose smaller pieces, smaller fragments by an enzyme called dicer. All right, dicer, it's an endonuclease enzyme which contains an RNase motif. All right, RNase motif. It's an endonuclease. It's an enzyme. It's an enzyme, endonuclease enzyme. All right. So what it does is it chops the double-stranded RNA into smaller pieces. So after now exactly the smaller pieces are 21 to 25 nucleotide long, and this double-stranded these small fragments are actually your double-stranded siRNA. Okay. Now how does this double-stranded siRNA works? Now this double-stranded siRNA, what it does, it incorporates with a protein called arginate protein, ARG, arginate protein. Now after incorporating with the arginate protein, what happens is that okay, let's get back to this. This double-stranded siRNA is actually, it has two strands. One is the guide strand, another is the passenger strand. This is suppose guide strand, and this is the passenger strand. So what happens is after the double-stranded siRNA incorporates with the arginate protein, this passenger strand just cleaves off, and now this arginate protein with the it just has the guide strand. Now it forms a complex called RIC complex, which contains arginate protein, incorporated arginate protein with guide strand, and this TR. There's another protein called TRP. So this whole together is called an RIC complex. RIC complex means RNA induced silencing complex. Okay, all right, inducing silencing complex. All right. Now this RNA induced silencing complex. So we know our target is this mRNA. All right, this single-stranded mRNA. What happens? This guide strand. It's complementary to this guide strand is complementary to that target mRNA. So now the guide strand actually leads the RIC complex to that specific mRNA. Now this RIC complex, what it does is chops or cleaves the mRNA and hence terminates the mRNA. So what happens is, if this mRNA is terminated, that means this is not going to translate any protein. No translation. So no translation means no. So this can be an advantage because we can use this as a weapon to stop the overexpression of viral proteins. At times viral proteins are overexpressed. So if we use siRNAs like this, siRNA in this method, then if we can specifically silence that particular gene, then that viral protein won't be expressed anymore. Okay. So now there are tools to there are bioinformatics tools or interfaces that gives you ideal candidates. Suppose if you go to that website called SiDirect2, SiDirect is one of the you'll just input your sequence, and then it will consider various parameters like obviously the size range, then the melting temperature, then repeats, how many repeats. Usually more than 4 would lead to self-complementary, and this will form a loop. Okay, I'll talk about the repeat palindrome repeat sequence and melting temperature and everything in the next videos. Now again, you have to consider the delta G also. So considering all this, it will give you ideal candidates. Suppose this S1, 2, 3, and then you can further go to the wet lab and check whether you can actually whether this actually works as a gene silence, whether it actually silences your gene. That's all from today. Thank you.